Hi students, welcome to HSC Chemistry and Module 8, Applying Chemical Ideas. This is video number 9 and we're going to look at the last of our techniques that's associated with the analysis and identification of inorganic substances, and that is atomic absorption spectroscopy. So you can see it's the same sort of deal that we've been looking at, conducting investigations and or processing data to determine the concentration of coloured species or metal ions in aqueous solutions, and this time we're focusing on the technique of atomic absorption spectroscopy, or AAS. So AAS is a technique that was developed in Australia. It's used to identify the presence and concentration of substances by analysing the spectrum produced when the substance is both atomized in a flame and vaporized, and therefore will absorb certain frequencies of light. So no different to what we've talked about before, just another application of the use of electromagnetic radiation to help in the identification of substances. Of course, specifically what we're looking at again are metal ions in solution. AAS is particularly useful for small concentrations, and this is where it kind of adds on to a lot of what we've looked at in the past. And you would have noticed just one of the previous videos with the calibration curve that we're actually looking at quite low concentrations now. This is really important because at low conc, precipitation often will not occur. So if we have a very low concentration of particular ions, and this is especially true for something, say, like lead ions, we know that lead in very low concentrations can still cause significant health effects to humans. And we know that we can precipitate lead chloride, for example, to identify the lead. The problem that we have, of course, is that the concentrations that we need in order to form a precipitate can sometimes be higher or much higher than the safe levels. So therefore we could try a precipitation reaction, say, well, there's no precipitate forming, and so therefore there are no lead ions. And in actual fact there are lead ions, they're just not enough in the solution to form the precipitate. So this technique allows us to detect very small concentrations, particularly of these sorts of metal ions that could have a damaging or detrimental effect if we find them in our environment. This is just a diagram that's not dissimilar to the one that we looked at previously, and as I say, you're doing a lot of the same sort of processes here. You can see once again we have our source of EMR. We have our flame, which is heating up our sample. So the sample will basically be sprayed into this flame or aspirated into the flame. There it will be atomized and vaporized. And so hopefully we'll actually be able to have our electromagnetic radiation hitting individual atoms. What will happen is then they will pass through a monochromator, and the monochromator is basically trying to extract just a single wavelength. Okay, so single wavelength. That will then go into a photomultiplier, just make the signal an amplifier, the signal, and then we get it as a display. The important thing about atomic absorption spectroscopy is that usually what we will have is specific types of lamps, specific types of emitters of radiation that are often made of the same substance as the substance that we're looking to test in the sample. The reason for that is that what we want to try and do is make sure that we select the wavelength that's going to be the most appropriate. So it's not just a single wavelength, it's the most diagnostic. That is, if a particular wavelength of electromagnetic radiation is absorbed by copper ions, and the same one is also absorbed by iron ions, then what we find is that the that particular one may not be as useful as a different wavelength which is only absorbed by the copper or only absorbed by the iron. So looking at spectra is very important to get an understanding of which of these wavelengths is going to be the most important, the most diagnostic, and therefore will give us the most information when we analyse the degree to which it has been transmitted or absorbed after it's passed through the sample. Just as we looked at for our other techniques in terms of UV-Vis and colorimetry, AAS also relies on the calculation or construction of a calibration curve. So once again, we need to determine we need to prepare a number of samples of known concentrations, which we will test first, and then we will be able to determine the concentration of our unknown sample. So this just gives you a little walkthrough of the processes that are part of AAS. Now, I can't see anything that specifically asks you to do this. What you will find if you look at some material from the previous course is that AAS was covered in quite a bit of detail. So you'll find this sort of a list of things. So our little cathode lamp, so the cathode lamp is going to correspond to particular ions that we're looking at in our solution. The light produced is passed through the vaporized sample. The degree of light absorbed is proportional to the concentration. So therefore, if the concentration of our sample is high, so the absorption will also increase. The intensity is measured, and comparing the intensity with control samples with different types of concentrations, we can start to build up a little bit of a picture of different solutions and their various absorbance, which we can then compare with our unknown. So this allows us to produce a calibration curve, and this is no different to the one that you saw before, and you can see this one has two different values which we have used our calibration graph to interpolate. So this would be a chart for calcium ions, and you can see for the calcium ions that we've got again roughly a straight line between the values that were taken from the calibration process, and then two unknowns X and Y that we've then been able to test with the same lamp, with the same setup, and then to be able to put them onto our graph and to work out what values we would have in terms of the concentration in parts per million. The setting up of a calibration graph is very important. It allows us to use an absorbance that we've got from our machine in order to then interpolate a concentration of ions that are present in the solution. Sometimes, because the ions are in such low concentrations, you may have to carry out one or multiple dilutions in order to get the concentration low. So if you carried out a 1,000-fold dilution, for example, in order to measure the absorbance, then you just need to remember when you go back to your original solution to multiply the value that you have by 1,000, as the concentration of the original sample would be 1,000 times that of the dilute sample. So these are some of the things that are associated with AAS. The most important thing to take away from all of this, of course, is that a spectroscopy is a really great analytical technique for identifying a range of different solutions, a range of different ions that are present in solutions. But more than that, we can use the techniques of spectroscopy in order to give us some quantitative data about the concentration of particular ions in solution. You will get an opportunity in class to prepare and present some of these calibration graphs for different types of ions, and also to use them to interpolate values for unknowns. Thanks for watching.